109 of the Stem Cell Podcast, Neuronal Cell Fate and Function, with Dr. Thomas Vierbuchen. Hello, everybody. This is Dr. Dalen James with Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotech supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have my boy from right down the street, Thomas Vierbuchen. He's from the Memorial Sloan Kettering Cancer Center, and he's going to talk with us about his research on neural development, amongst other things. He's got a really deep lore surrounding his work that's going to be fun to dig into. We've also got our usual roundup of recent highlights in stem cell news. That's coming right up.
But first, looking to incorporate neural organoids into your research, get a free wall chart showing how neural organoids are generated and can be used to better understand brain development. To learn more, visit stemcell.com/brain organoids-wall chart. And yeah, it's going to be fun to catch up with Tommy, a fellow Stanford PhD alum, and he was a couple years older than me, couple years my senior in the PhD programs at Stanford, but he did such incredible work during his graduate training and has continued to do incredible work many years later in his own lab. And we're going to start things off with something that's in the neuro adjacent area, I suppose. I mean, we're talking about spinal cord injury and spinal cord regeneration. This is a Nature Biotech paper, and we've talked a lot about the history of this field a lot on the show. Initial Geron trials and all that kind of stuff for the promise of using embryonic stem cells for repairing spinal cord. That was decades ago now, right? And this is not a direct progeny. It's inspired by that, I'm sure, in some capacity, but it's related, right? We're talking about work that's coming from Mark Tazinski's lab down there in San Diego, but has had it's been many years in the coming here. This is a non-human primate system that's showing non-human primate model that's showing extensive restoration of forelimb function in these primates that have undergone a spinal cord injury or SCI. And the key here is using embryonic stem cell derived neural stem cells. So NSCs derived from H9. So we'll get into it. This, like I said, this has been a dream in the field for a long time now. One of those critical hallmark potential cell therapies for pluripotent stem cell derived cells, and it's been sought for many years, but it's yet to achieve sufficient functional success and recovery in animal models, and of course humans as well. And so they're starting here with the animal models. Many years of mouse work and in vivo work leading to these non-human primate studies. Previous studies in the field from other folks have grafted oligodendrocyte precursors and progenitors, non-spinal neural stem cells, different approaches, including like primary spinal neural progenitors, right? But here they're seeking to improve functional outcomes in spinal cord injury in this non-human primate model by using clinically compatible spinal cord neural stem cells, importantly derived from human embryonic stem cells, the classic H9 HESCs. So these are H9 derived SC-NSCs. That's what they call them. H9 derived spinal cord neural stem cells. And when they grafted these things into these non-human primate models, they showed that indeed there was significantly improved functional outcomes on a skilled hand task, behavioral study, 9.2-fold improvement actually in these hemisected subjects compared with the lesion controls. So they had a dramatic improvement in the fine object retrieval success using their hands of more than 50%, plus or minus 20%, and close to 3-fold superior to their other controls in hemi-contused subjects, so the partial control here. So functional recovery to some extent in these monkeys, which is really cool to see. The recovery also correlated with the rehab effort and the graphs. When they took a deeper dive into the histology, they showed that the graphs actually extend up to hundreds of thousands of new axons into the host circuits, which are actually 4 centimeters below the injury. So these new neurons are extending new axons deep into the host tissue, which is very cool. I mean, that's the goal here is to restore that motor function. You got to have that extension and reintegration below the injury site, and they're forming synapses with the host circuitry. So there you go. And they did a bunch of imaging histology to confirm that. And they also showed that the filling of these lesions was a lot higher than had been demonstrated before, um, and differentiated cell fate distributions were actually a lot closer to that of the normal spinal cord. So you're getting a restoration somewhat to the normal phenotype. So they present this as a dramatic improvement over prior studies that were using, for example, primary spinal cord cells. And part of this maybe has to do with the malleability of HESC derived, neural stem cells, these earlier progenitors, more plasticity, right, in their fate and ability to generate new neurons and extend their axons deep into the host tissue. So this is I think, a very promising result, right? It's a Nature Biotech paper, really promising functional results in these non-human primate studies, which are never easy to do, as we've talked about here on the show. But um, hopefully, this is enough ammo for them to move towards clinical trials.
Yeah, this is so good. So good. The story behind the story, the story behind the science is great. And just to age myself quickly here, my first ISSCR meeting that I went to, I can recall that Hans Kierstead went up and he was talking about the spinal cord repair, and he put up this video. He put up this video, which was actually quite advanced as like even like animation at the time because it was all 3D rendered, and it was a video of these rats like crawling along the surface, um, to kind of illustrate the potential. And he was like, "We're doing this," and then everyone. And by the way, in the moment, everyone was like, "Oh my god," freaking out. And then he got blasted, of course, because the story wasn't published. It wasn't like peer reviewed published. He was just showing like preliminary data. And he did show, in fairness, he did show like some actual mice, not just cartoons. Um, but everyone went nuts, and then everyone went after him. And everyone then with the Geron thing, as you mentioned, like everyone was like, "Oh, wait." And I think it's a great lesson, put into the framework of caution at the forefront. Maybe learned a bit from the gene therapy trials, which we finally recovered from in, massive scope. Um, but it was too quick. And here we are, not even in a rodent, right? In a primate, which is so, and as you said, the extension of these hundreds of thousands, like we're talking real scale, like human level, primate level repair, not to mention all the functional stuff, which of course I think there's a lot more improvement, maybe even in these monkeys that could be demonstrated over time, but certainly as the techniques improve. But the best part of the story for me is the H9. I'm glad you underscored that, because to me, there's like a real poetry in that. Like, as far as we've come in IPS and organoids and everything, we're still using this first line, founder line in the most advanced approaches, which I think is a testament to and a real delivery and affirmation of the amazing promise of stem cells. Like maybe Jamie Thompson should get the Nobel Prize. Did they talk about that? Because this is great. Him and others. he and others. But I don't know. Such a great story. So many stories that kind of culminated into this. Uh, so poetic and really impressive, exciting, scientifically, clinically, all of it.
Yeah, absolutely. The the OG H9 derived decades ago is having its day in the sun here. And it's not just this study. There's, I think, a resurgence in the use of HESCs and the H9 derived cells for various clinical applications that we've talked about here on the show. And I agree with you. I mean, you don't have to ask me whether or not Jamie Thompson deserves a Nobel Prize. And I know there's some politics behind all of that, as it all is with the Nobel these days. But I mean, yeah, looking at this particular study, the improvement is pretty dramatic. And I mean, one thing I think you're alluding to is following these animals even beyond the scope of this study. I think they only looked at them for about half a year in terms of their functional improvement. So I'm curious to see how this keeps on going and if this is really a stable engraftment and the functional improvement is also stable.
Yeah, man, this is so cool. What an exciting time to be. I got to say that at least once a show. What an exciting time to be in this field.
Yep.
Speaking of exciting, I mean, here we're going from the point of the spear to the staff, maybe. I don't know if that's an apt analogy, but I'm talking about here a field that I'm, you know, a big fanboy for, which is hematopoietic stem cell transplantation. And I think a good counterpoint there to these, spinal cord repair, which again is cutting edge here, the tried and true as well established therapy in the stem cell field is hematopoietic. Um, and it's straddling both the history, right, of just garden variety bone marrow transplant and cord blood after that, now to the cusp and the cutting edge again of gene therapy, right, where you have sickle cell, also CAR T. So it's really such a core element of the clinical translational application of stem cells, but of course, it's hampered by the inadequate doses, really, when you're speaking clinically, which underlying that is that we're unable to self renew and maintain and expand hematopoietic stem cells in the new therapies, genetically modified hematopoietic stem cells, without compromising their functional properties, right? Even in the trials that I'm alluding to there, like the CRISPR-Cas genome editing for sickle cell, for example, the patients required between 3 and 18 days of apheresis after mobilization of the stem cell compartment to get enough cells, right? And that's not to mention the whole idea of cord blood is predicated on the idea, or cord blood expansion is that one cord's not enough, and donor marrow and HLA matching is limiting all these things, right? Um, so the one underlying limitation, and granted, there's been a lot of approaches, and I've talked about almost all of them, and every time I'm like, this is it. And guilty again. I think this might be it, but I'm getting ahead of myself. Um, the one fundamental issue I think with all of the approaches in the past, and we're talking about the small molecules or the ex vivo kind of niche support, have really focused like fundamentally on either preventing differentiation or maximizing quiescence, which is like sounds like the same thing, but actually there's a subtle difference in approaches for how you maintain the stem cell pool. Um, enter the lab of Vijay Sankaran, who's at Boston Children's, whose lab recently showed that in the context of bone marrow failure in humans, the hematopoietic stem cell pool displays this unique vulnerability to loss by ferroptosis. So here, so the Nature Cell Bio story, they went after this to resolve this in a more clinical translational context. They kind of validate that approach first, showing that the HSC loss in vivo or ex vivo in these expansion platforms can be largely attributed to ferroptosis, um, which, the pharaoh, I mean, if I'm pronouncing it wrong, I'm embarrassed, but not really. I mean, that's a tough one. Uh, Tomato, tomato. But the pharaoh there is a iron element. It's iron dependent cell death pathway. And because it's iron dependent, you can block it. And so that was the approach here that the Sankaran lab took to augment hematopoietic stem progenitor cell expansion. They used either liproxstatin 1 or ferrostatin 1 and showed that it markedly increased the expansion of cord blood and adult hematopoietic stem cells in all the medias, not all of them, but the representative medias, both serum free and chemically defined conditions. And then they showed that these cells, after expansion for, I think it was a week, and maybe I'm shortchanging them because maybe they did longer ex vivo expansion, but I'd be interested to see if they could go longer than a week. Not a critique, just curiosity, because really a week can be sufficient, especially if you're getting expansion to mediate this genome engineering, right? So this is it. There I go again. This is it. But they show that these cells can then, after expansion, and can uh, durably engraft multi-lineage contribution in xeno transplanted mice. Going into the mechanism a bit, they show that ferroptosis blockade is the way it does it is by increasing ribosome biogenesis and cholesterol synthesis. And because of the increased cholesterol, you get increased levels of this metabolite, 7-dehydrocholesterol, which in itself is a potent endogenous ferroptosis inhibitor. And that in its own can promote hematopoietic stem cell transplantation. So, I mean, as I said there, this weak ex vivo expansion, while there may be a lot of challenges in terms of like, well, hematopoietic stem cells need to self-renew for the lifetime of an animal, blah, blah, blah. Clinically speaking, this is a entry point to all of these, like we're talking thousands, or if not already imminent genome engineered hematopoietic progenitor stem cell therapies, like all, I think, will be facilitated and, uh, meet their endpoints more likely because they have a platform here which is reliable. Arun, is this it?
Maybe? That's my, uh, politically correct answer. Maybe. Um, no, I think it's another tool for the toolkit here, and the reason I like this story is because it's so easy to do, it's the just they just say it, right? It's so easy that you can put these antioxidants or these ferroptosis inhibitors into whatever media you want, and it seems like it was universally beneficial. So the tool builder in me and the differentiation tweaker, protocol developer, whatever you want to call it, I love these kind of papers because it's just something that you can immediately take and implement into your own cell culture or your own expansion profiles if you're an HSC biologist. And I'm sure folks have immediately started doing this, buy a small molecule inhibitor of ferroptosis or an antioxidant treatment, add them to your own cultures. You can do this within a few days. Um, and, we, like as you're alluding to, we've talked about these stories in the past, other promising aspects of or promising approaches to improve expansion of HSCs. And maybe it's just a matter of developing the right cocktail and, having these ferroptosis inhibitors. Maybe that's a critical element.
I'm getting the pronunciation in. Yeah, yes, totally, totally. So easy, so basic, to the point almost where you're like, really? How can anything be that easy? Too good to be true. And the cynic skeptic in me is like, all right, let's see, let's see. But I want to come back to that for two reasons. One, because I think that the maybe the keystone here is this novel approach, right, of targeting ferroptosis. I said it. And that it's a novel angle, which then the cynic in me is like, but really, like in these on these massive screens that people have done, none of these relatively basic compounds were a hit. But whatever, that's my inner turmoil. You don't need to be bored with that. The other side of that that I want to emphasize is that I love the story here of tracking from an observation in a human patient cohorts, right? And I love in a place like Boston Children's is de rigueur, but like, I think it is really the best kind of science that follows from observation, yeah, sure, but like patient observation. I'm always so envious of those MD PhDs who are real, bench scientists, but also are so informed by their whatever 2 days in the clinic each week because they really understand the problems and have kind of all the angles. So I love the way they came about it in this case.
Yeah, and I think Vijay Sankaran is that hallmark quintessential physician scientist, and I think is a perfect fit for what they're doing over there at Boston Children's. And he's been a world leader in this field for a while now. So shout out to their lab. Moving on to something that's, near and dear to my heart. I don't know how many times I've made that reference or made that joke, but you know what? I'm going to do it again. So here we go. Cardiomyocytes, heart biology. And, for frequent listeners of the show, you know that one of the reasons why I got into heart biology was this idea, this dream of being able to regenerate the human heart. And back at undergrad at Duke University, go Blue Devils, I didn't work in the lab of Ken Poss, but I got to know him pretty well. And of course, his work has this, he's shown this amazing regenerative capability of the zebrafish heart, which is lost in mammals. Now, the subcontext of that story is that regeneration of the heart isn't entirely lost in mammals. It's just not there in the adult. It is there to some context in neonates, and it's lost pretty quickly over the course of postnatal development. And so that's kind of what the foundation of this study is about. This is a paper coming from An Zhang at the University of Chinese Academy of Sciences in Shanghai. Cell stem cell paper titled Injury Induced Clusterin Positive Cardiomyocytes Suppress Inflammation and Promote Regeneration in Neonatal and Adult Hearts by, the important part here, is reprogramming macrophages. So, exactly on that topic, right? Adult hearts, adult mammalian hearts, human hearts scar after injury, but, and it's been known for years now through a lot of, detailed mouse work, that neonatal hearts regenerate, just like zebrafish hearts regenerate, right? Now, we're still trying to get to the bottom many years later of why that's the case. Why do you have that regenerative capability in the neonatal heart? Why is it lost pretty quickly at birth? And here they did a bunch of single cell analyses, spatiotemporal analyses, lineage tracing, of course, and saw that there is an injury induced clusterin positive or clue positive CM cardiomyocyte population that coordinates what they say as a reparative anti-inflammatory macrophage activity. So you're getting a connection here between the cardiomyocytes, the muscle, the functional muscle cells of the heart, and those immune populations, those resident macrophages in the heart as well. And they're showing here that after injury, these clusterin positive cardiomyocytes emerge in that injury border zone of regenerative hearts, but are not there in non-regenerative hearts. So they're found in those neonatal populations, neonatal hearts that are undergoing regeneration. And so they took a deeper look into what's the deal with these clusterin positive special cardiomyocytes. And they saw that they indeed secrete CLU, clusterin, which is a protein that binds to macrophage toll-like receptor 4 or TLR4, which actually blocks inflammation, promotes reparative polarization through fatty acid oxidation. So they got deeper into the mechanisms there. And it's really the macrophages here that's the key. These macrophages are secreting their own BMP2, bone morphogenic protein 2, one of the TGF-beta superfamily proteins, which activates, as you would expect, TGF-beta signaling, BMP signaling in these cardiomyocytes. So there's like a crosstalk going on um, to drive the proliferation ultimately of the CMs. We know TGF-beta signaling is very important for regulating cell growth, cell proliferation, a lot of different things. And here, it's these macrophages that are secreting BMP to induce proliferation of these myocytes in the regenerating heart, in the neonates in particular. So the they actually took this to a very interesting level here in a clinical level, which is really cool. It's sort of the tail end of this paper, where they show that in patients, so patients with MI, myocardial infarction, if they have reduced CLU levels, it actually correlates with impaired cardiac function. I think that's kind of a stretch there because, of course, as we know, adult human hearts don't regenerate. But I think they were trying to make a parallel to the importance of the CLU protein as kind of like a biomarker, right, for cardiac function. And I don't think they're trying to say that, you know, there's a human adult repair element here, but I thought it was maybe just like an interesting kind of sprinkle on top of the cake for this mostly basic science study. Now they took it. I really love the tail end of this story because it was very cutting edge. Because not only did they look at that biomarker for CLU in cardiac function, improved cardiac function in these patients, they actually over expressed CLU or transplanted CLU positive human cardiac organoids into their mouse model to actually show that when you do that, you improve the regenerative capacity of adult mice, which is pretty cool. That's a pretty cool approach. So they took an observation initially from development in neonatal regenerating mouse hearts. They saw maybe that CLU is a biomarker in humans, and then they did the cool organoid engineering and saw that CLU overexpressed organoids can actually improve regeneration. Yeah, not in humans for sure, but adult mouse hearts. So there's this cardio immune crosstalk mechanism, and this has become, I think, a really really hard topic, really hot topic in cardiac biology. The importance of the immune system and regulating cardiac function, and this is just another example of that, of how macrophages might be the key here to cardiac regeneration. We'll have to wait and see.
Yeah, this is it's got so many elements. I'm glad you mentioned that last part. I remember it was like 10 years ago where they started talking about, CHIP, clonal hematopoiesis, and the impact on heart function and monocytes. And it seems like, yeah, that combined with this neonatal cardiac repair phenomenon, all those things are Frankensteining up into this very, I would say, clinical. I know it's basic, sure, in terms of the biology, but I think what this is bridging is that notion and what you said, that last part, which I agree is probably a more exciting and technically complicated and cool part of this paper. I think that was the key to getting it through here: cell stem cells, human relevance, clinical relevance. Um, because yeah, that's kind of the thing. I don't want to say the story's grown stale, but I think that there's been this stall of all right, neonate. It's a phenomenon in neonates, but how do you then endow the adult heart, right, with that? It's not just a matter of the signaling, but the scale really, and crossing from maybe mouse and zebrafish, scaling up to human. I don't know. It's just been tough, heavy lift, right? Um, so that yeah, that's where I think this story is really impactful. Is that it bridges that divide and kind of raises the stakes a bit. Um, and yeah, the immune component, as you said, very cool to come full circle on that chip idea. The last thing I would say is that the mechanism here in terms of BMP signaling, you said it, TGF beta, it's the target probably number two, right? After Wnt, I'm sure. Everyone loves Wnt. But PGA beta signaling, like, it's so implicit in cardiac signaling that BMP, there have been so many attempts to hack the axis in efforts of post-injury, peri, whatever. So and I'm not like throwing shade at all. What I'm saying here is that because maybe they did try, but maybe a reviewer asked, like, what if you just add BMP? Like the truth is that it's not enough to just dump a recombinant or whatever kind of cytokine secreted factor in there. It really is about the nuanced and paracrine, direct, juxtacrine, whatever it is, the orchestra of signaling locally and dynamically that governs these processes. So what I really like about this story is that it's kind of like this, like by way of a local, conductor here, the macrophage. Like I think that seems more viable in terms of how we're ever going to translate heart repair. Like we can't just throw cells or cytokines in there. Like we need to get an organizer in there to kind of take over.
Yeah, and I think there's a lot of cool elements to the story. Um, the other part of this, and correct me if I'm wrong, I really haven't seen the cardiac organoid model as a cell therapy in itself, right? And this is because most of the time when you think of organoids, you think of an in vitro model for modeling development, disease, drug screening, all this kind of stuff, but they directly use these cluster and overexpressing cardiac organoids as like as the therapy itself and as like a delivery vector for clustering. That's cool, and I'm curious about what like Chuck Murray might think about this when it comes to the feasibility of using organoids themselves as the cell therapy as opposed to these engineered heart patches or genetically modified cardiomyocytes that he's working on in his lab. And very last note is that these were not iPS-derived organoids. They were created from our old friend H9. So H9 derived
Star of the show.
star of the show here today, Mr. H9 OG embryonic stem cell line. It's still getting its day in the sun here.
Deserves it. Been a long time coming. Yeah, you're right there. That kind of good segue to my story, or which is an organoid story, and I'm going to come back to this maybe in the end. Is like I don't know if you're getting at this or not, but like I feel like clinically, therapeutically, scalably, industrially, like maybe the inputs need to be a little bit more defined, and that's kind of what I'm going to talk about here in this. Staying in the theme of blood. If you get to talk about the heart, my man, I get to talk about the blood. So I'm going 2 for 2 today on my end. This is like more than the blood, though. This is all the other stuff surrounding the blood. It's a niche story. And of course, we all know about the bone marrow niche regulates blood production. And that's I talked about earlier the difficulty in balancing the self renewal and differentiation and maintenance, homeostasis and metabolic signaling, migration. There's so many things that decisions that a hematopoietic stem and progenitor cell needs to make over the lifetime of an organism. And there's multiple niches, and it's been subject of hot debate. Sinusoidal, perivascular. Here we're talking about this endosteal niche, right? Which is complex in and of its own. It's in itself. It has multiple other components, including the vasculature and osteoblasts. This is the bone. This is at the surface of the bone, but also neural cells, and of course, you have all these immune cells trafficking around there. Um, so why are we talking about the industrial niche? Well, because it does a lot of things. It's involved in a lot of like homeostatic processes, like stress-induced mobilization, regenerative hematopoiesis, but also there's a ton of bad news stories involving the industrial niche in terms of like development of malignancies, or like chemotherapeutic resistance, or it's a site for solid tumor metastasis. Stories about the aging endosteal niche and how that underlies like maybe these myelodysplastic syndromes and other kind of monocyte dominant disease manifestations, as well as the poor bone maintenance, osteogenesis. Um, and although it's involved in all these bad news elements of human health, uh, we don't really study the human endosseous. We have a ton of studies in the mouse, which are, honestly are mostly centered on this site for hematopoietic self renewal. But we got access, at least, to the endosteal niche in mice, but in human, really poorly investigated. And no coincidence, because it's like, yeah, it's I don't say it's like the brain, but it's tough. It's tough to get access to any tissue, and in this case, no different. And it's hard to when even if you can get it to reconstitute, the that niche to maintain it as it is when you take it out. It's like a real bioengineering challenge, right? So this is a story straight out of Basel from Ivan Martin and Andres Garcia, who are at the University of Basel. And this is a big splash. I mean, we don't read a lot of stories of Basel. They're doing their stuff over there, as represented by this big story where they're making bone marrow organoid upgrade models, right? So there's like a bone marrow organoid models that have been published in the past, but and they're good. They can recapitulate both healthy and malignant hematopoiesis, but they don't have any bone compartment, and that's real because without that superstructure, you get these like micron level bone marrow niche models, which don't recapitulate that kind of sinusoidal vessels, or at least I don't want to say sinusoidal, but scale of the vasculature in the endosteal niche. And also, those models depend on matrigel. So here, um, they upgraded and they made this macro scale, right? So way larger, orders of magnitude, not orders, but an order of magnitude larger than the existing bone marrow niche model. And they include this endosteal component. They called it engineered vascularized osteoblastic niche. Clean, a nice clean Swiss acronym, EVON. And they have vascular networks that are covered by parasites. They also have neural fibers, and it's all embedded in this osteogenic matrix. They have a lot of the signaling components, namely SDF1, KIT ligand, also VEGF. They're expressed in patterns that are native to the human, um, the human endosteal niche. And then they can get supportive hematopoiesis in vitro and then transplant. So similar to, I wonder if you combine this with that Sankaran story, because they get multilineage repopulation capacity after carrying these things, cells, primary cells in this endosteal niche. And then they do some modeling. I guess they had to do some modeling to get into cell stem cell. So they do these perturbations where they blow out the vasculature, or they inhibit VEGF signaling, or CXCL12 with SDF1 signaling, and they show that you need those things. You need those things for myelopoiesis. So a cool story, I think, a clean platform. As I kind of said in the beginning, this and the cardiac organ is more defined, right? Because I guess the inputs they have to sort, but maybe not. I am a little bit wary of these. Not, I feel like you got to define the input. You got to get a clean population and say this is what's going in order to have a really clean model and to have complete like mechanistic understanding of what's going on in there, which this doesn't necessarily have. These endosteal organoids are like really complex, have a lot of other cell types in there, but still the function I think speaks for itself, and the pictures are really cool. You see all these different markers that are all overlapping and interconnected, speaking to just the power of this native embryonic potential. A little shout out to H9 again, going back to the future. These cells can do a lot of things all by themselves with a little bit of guidance, and this is a great tool, another tool that we're highlighting here in the show today.
Yeah, another awesome tool for the toolkit. I feel like we've had a very diverse range of stories here, ranging from organoids, scaffolding, engineered approaches, cell therapies, a little bit of everything, reflecting the diversity of the stem cell field and when it comes to the topics, right? So here, yeah, it's more on the engineering side of things with this 3D model. They only focused on really the niche itself, these stromal cells, and they say on the technical side that they still gotta optimize their digestion protocol that actually was used to obtain that single cell suspension. in their words, not optimal for efficient cell retrieval from those scaffold pores and also for preserving the endothelial cell fraction. So maybe that might explain some of the results here. One other thing to mention here is this was a very heavy single cell story. And on the paper, I think one of the senior authors is Barbara Troutline, who, of course, is an expert in all things single cell. I think received an ISSCR award for momentum or early career a few years ago. So I think a really interdisciplinary story here coming from beautiful Switzerland.
Yep, that's for sure. They got it all over there. Kind of had it all in these organoids, like you said, interdisciplinary, intercellular bonanza. And we're gonna go from the core elements, and I like how you talked about the diversity, all anchored by the real the fundamental ground truth of science and life, really, which is H9 and hematopoiesis. Everyone, I'm sure you'll agree. We're gonna go back a little bit more into history in just a minute with Tommy, who's gonna tell us. I mean, we're gonna turn the screws a little bit on his graduate school experience because that's a piece of history, and talk about other things, both basic and clinically applied. So, exciting things to come. Before we get there, I got a quick message from STEMCELL Technologies. Neuroscientists looking for more predictive power in their disease models are increasingly adopting human pluripotent stem cells in their research. STEMCELL Technologies offers products, protocols, and training to support HPSC-derived neural models. Explore their collection of technical videos and webinars on neurological disease modeling by visiting www.stemcell.com/neural disease model. All right, everybody. On today's episode, we have with us a special guest from right down the street from me, Dr. Thomas Vierbuchen, who is Assistant Professor of Developmental Biology and member of the Center for Stem Cell Biology at Memorial Sloan Kettering Cancer Center. The Vierbuchen Laboratory studies the mechanisms governing the production of the myriad neuronal cell types required to build functioning neuronal circuits. To model these dynamic developmental processes, lab members direct the differentiation of mouse and human pluripotent stem cells into specific neuronal cell types in a dish. In the long term, they'll apply these insights to better understand the complex genetic underpinnings of psychiatric and neurodegenerative diseases and conditions. Tommy, thank you so much for joining us on the show today.
Thank you. Excited to be here.
We are very excited for this conversation. I mean, we don't talk enough or really at all, considering our proximity. So it's a shame, but we're going to get a jump start to that conversation, catch up here, and move forward. Why don't we start with you giving our listeners a summary of what your group's working on right now?
Yeah, so my lab is interested in understanding the development of the brain, and we use a variety of model systems to do that, including mouse and human pluripotent stem cell models in vitro. And we're really interested in understanding how the genome orchestrates the process of brain development, which is incredibly complex. And that requires very sophisticated mechanisms of gene regulation to turn the right genes on and off at the right time and place to produce the diverse neuronal cell types that are necessary to make a functioning brain. And so we have been mostly developing new models starting from mouse pluripotent stem cells, where we can generate neural organoids and other neural cell types in order to be able to understand these genetic programs and how they vary between individuals and also in between different species.
Yeah, I mean, back in the day, Tommy, and you've been in the game for a while now, we're all just so excited just to make these things in 2D, right? 2D neurons, or as a cardiac biologist, 2D cardiomyocytes. Just making them was an accomplishment. But there's been, like you alluded to, such a dramatic shift in the differentiation protocols and complexities, especially when it comes to the 3D organoid differentiation approaches, for example, cortical organoids, assembloids, and so on. I mean, But even so, we've reflected recently on the importance of the 2D cultures and dissecting some of these cell intrinsic phenotypes and neurons and in cardiomyocytes alone, for example. So even though there's been that dramatic evolution in differentiation protocols, could you reflect a little bit on, the old school approaches, some of the 2D approaches, and whether there's still a utility for some of those simpler differentiation approaches?
Yeah, for sure. I think the 2D approaches are still incredibly important. Because organoids are still they're very complex. They're difficult to generate reproducibly from large numbers of cell lines, and it's really it's not easy to analyze an organoid as well. Like when you want to look for a phenotype, you often need things like single cell RNA sequencing, or you need dissection, do staining. But the 2D setup can be extremely useful for looking for large scale phenotypic screening and other questions that are not really easily answered in an organoid model. And so I think you're right that nowadays it's all about organoids, but I think there's really all of those different approaches have important roles. I think in the range of things that people want to learn about developmental biology and disease. And so I think, especially industry is very interested in having sort of more homogeneous 2D models, and that's where things like, NGN2 neurons are, very widely used because they're relatively easy for people who are not stem cell biologists to sort of import into their labs. And so I think the evolution in the field has been, people that used to study kind of, neuronal cell lines that were kind of mysterious origin now can generate actually human neurons in their own lab and do basic studies of, cell biology, gene expression, whatever that lab is interested in. Instead of doing that in sort of a strange cell line, they're able to do that in a kind of a nice human neuronal cell type. And so I think that's been very transformative for labs that kind of have questions that are related to molecular and cellular things. They don't care as much about the developmental biology. They want to just have cells that they can work on. And that's where I think those 2D models have been very important for the field.
Yeah, I mean, I think about that a lot lately with as we're like reaching the kind of apotheosis of our field here, modeling true human development. And I reflect on that like there's almost like we're in a race to get there. Like that's the goal, but really, all of these things are tools, and really, maybe the most impactful tools are these really simple models where we can ask really big questions in a way that's scalable and repeatable. And not to mention, there's this fixation on the 2D, 3D, 2D, 3D. But like the way I recall, because I'm an old fogey pretty much at this point, is the very first thing we were doing was making EBs and then putting them in adherent culture. So like that's not really 2D. I mean, I know there's a lot of monolayer in our field, but like anchored 3D culture maybe is what you would call it. Sometimes is the ticket. I find in my organoid diffs, they're very static. It's hard to get them to move forward. It's hard to get a lot of proliferation, but I'm digressing a little bit, telling my story. Let's get back to you, Tommy. Regardless, putting into context, you can see how the semantic element over these long scientific arcs play out, like 2D, 3D. And for me, it's really gratifying to have been doing it long enough to see some of these, big kind of field shifts, tectonic shifts take place. And one of those was directed reprogramming, right? Of terminally differentiated cells into defined fates. And you were the guy, first author on one of these, arguably like the initial bombshell study back in 2010 with Marius Wernig, cited almost 4,000 times to date. Congratulations. I mean, that's amazing. What was that like, being at the center of science for a spell? And where are we now, in your opinion, with this direct reprogramming and conversion?
Yeah, I mean, so that was a really exciting time in science in general, not just for me, but it was Marius started his lab at Stanford in the fall of 2008, and he was coming off just making, probably the second person in the world to make iPS cells after, hearing about Yamanaka's work. And, so there was a tremendous amount of excitement around reprogramming. Papers are coming out left and right, making iPS cells from human. And I think, that was really what excited me about joining Marius's lab at Stanford and so I was the first student who joined his lab. Actually, it was, just two technicians in the lab and me at first. And so, yeah, so I mean, I think, we wanted to be able to generate, Marius had this idea that basically, if you can reprogram somatic cells into pluripotent stem cells, it could be that you're essentially erasing all of the epigenetic memory of a cell and going back to some default state, which is pluripotency. And that might be like a unique example where reprogramming is only possible to the pluripotent state. But he, had this thought that, well, maybe, what if we tried to make, something like neural progenitor cells, which are, developmentally somewhat closely related to pluripotent stem cells because they come directly from the epiblast during development. And people had at the time shown that neural progenitor cells can be more reprogrammed to pluripotency with only 3 factors instead of 4. So again, the idea being that maybe they're closer already to pluripotency, and so it might be possible to then actually take fibroblasts and reprogram them directly to neural progenitor cells, was sort of the thought. But the way we set up the experiment, it ended up that we had proneural genes like ASCL1 and neurogenin in a different set of factors that we were introducing into the fibroblasts. And what we saw in those initial experiments was that we saw cells that look like neurons in the conditions where we overexpress the proneural factors. And so even though that wasn't initially the goal to make neurons, we basically ended up, because of the way we set up the experiment, getting these postmitotic neurons instead. And so then we ended up following up on that. And that was a sort of a whirlwind year of, because it was very scary, really, because it's like such an exciting possibility, but you can also imagine a variety of ways in which it's maybe somewhat artifactual, or, are we sure, this is right? How do we know that the fibroblasts don't have neurons in there somehow? And so we had to do a lot of experiments to kind of convince ourselves that this was a real and robust effect. And so it was very exciting, but also very stressful. And, and then once the paper is out there, I think you hear people saying, oh, maybe I heard that's not so reproducible, or, but then, like luckily after, about 6 months or so, we had, other people reporting back to us, we see this working as well. And, that was really, a good time to be able to relax a little bit. But, we then also went on to try and do the same thing with human fibroblasts, which turned out to be a bit more difficult, but we eventually figured that out. And then similarly with human pluripotent stem cells, which led to the development of the NGN2 neuron protocol. So yeah, it was a very exciting time. And Marius is, I'm really glad, of course, that I ended up working with Marius, because it's just a great experience to have had as a PhD student.
Yeah, incidentally, we had him on the show recently, and he really had us laughing, because we asked him if he ever got back in the hood, and he was like, "No, not really, but I can do it."
Yeah.
Yeah, no, I mean, that was such an incredible moment, and I can only imagine your feelings when you actually saw those cells in a dish, those cells that look like neurons, were like, and you said to yourself, "Did we actually do this? This is so incredible." And, being able to be at the forefront of some of those observations as a graduate student, of course, is
Yeah.
another huge, milestone, huge thing. I mean, that's the dream for any grad student, right? To be involved in such a amazing story and make an incredible discovery like that. And like what we were talking about before the show, that paper was actually really formative for me when I was deciding what to do with my life as an undergrad. And it's a big reason why I actually decided to apply to the PhD program at Stanford in stem cell biology, which I'm assuming that you probably wouldn't been in the program if it existed when you were around in grad school, but that started in like 2012. So, and I also remember Deepak Srivastava, pioneering a similar direct reprogramming concept with cardiomyocytes. So, it was really just an incredible milestone. So talk a little bit about the vibe there at Stanford and Marius's lab that really enabled studies like that to happen. Of course, like you mentioned, Marius was one of the first folks to make iPSCs, and I think there's an advantage to being in a small lab initially as the guy, one of the few students in the lab. But tell us more about the environment that enabled that study.
Yeah, I mean, so I think Marius is a very creative scientist, and I think he really wanted to go after big and important things. And so I think, you know, like one thing he said early on when we were working on it, is something along the lines of, I forget the context, but he said basically that there's no speed limit on the autobahn,
Mm.
which is this. It kind of we sort of like always laughed about it in the lab, but what I think what he was trying to say essentially in his Austrian way was that you need to, there's nothing stopping you from going after big questions, and so I think that was the environment at Stanford at the time. We were actually located off campus in a little office park next to actually the Tesla offices at the time. On Arostradero Road, and it was basically all the new PIs who were going to be the stem cell center in the new stem cell center at Stanford, but they were had moved from other universities, and the building wasn't built yet. So Irv Weissman had half his lab there. Rene Rejo-Pero was there. Phil Beachy was there. Tom Sudoff was there. Rob Malenka. So we had actually not only stem cell biologists, but also some neuroscientists who were going to move into the center. And so that ended up actually being really important, because one of the key things that we had to show with the directly reprogrammed neurons is that they're actually exhibit features of neurons. And then we had Tom Sudoff's lab and Jipeng Pang, a postdoc in his lab at the time, who was able to help us show that these neurons have electrophysiological properties that make sense. And so it was really an interesting environment being over there, and we were off, isolated a bit from campus. And it was, of course, great once we moved into the brand new shiny stem cell building. But I think it was really, the California dream-in sort of vibe was very different from what I was used to on the East Coast. And so I think Marius, adopted that mentality very quickly, which is sort of like an anything is possible kind of environment. And so I think that kind of helped as well. I think, it's better, or it helps you, feel like anything is possible, really. And Stanford is such a great university. And, so it was really, I think, a special time. It's still great, but, especially with IPSLs in the air and, CERM funding and all that stuff, it was really, I hope we can recreate that again.
Yeah. I mean, when you think about it and hearing you talk about it, maybe that was the golden age of just stem cell research, right? Because now we're in this age of the industrialization application, which is maybe it's more exciting. But back then, as you describe it there, what a great tableau, right? Just people on the fringe, but the world, anything is possible. Everything's just been unlocked. You got all these talents and techniques laying around. What a great synergy and a lasting impact from that. So thanks for sharing. But you came back here to the East Coast, sucker. As winter approaches, I'm really feeling for both of us. But we got our own thing going here in the Tri-Institutes and the city at large. And what you're doing here is highly translational with clinical endpoints like neurodegenerative and psychiatric conditions. But you still got a little piece of that old school stem cell research from the golden era, basic biology part of your lab. You had this recent story in DevCell that went a bit afield, you could say, of neural, but maybe not. Make your case. Focusing on OTX in this elaborate circuitry. As you kind of said in the jump about what your lab's working on, it's really about the circuitry, right? in this case, underlying endoderm commitment during early diff. How does this study fit in with your larger goals and research focus?
Yeah, so that's a great collaboration with Matthias Stadtfeld's lab at Weill Cornell, just across the street. And so we one of the things that I was really interested in getting set up when I came to Sloan Kettering was developing improved differentiation models starting from mouse pluripotent stem cells, because at the time human protocols for directed differentiation into a variety of lineages were starting to get better and better and more highly optimized. And that's great. And it's great to be able to work with human cells. But a lot of people don't necessarily need or want to work with human cells. They just want to work with cells that have the right transgene or the disease mutation that they're studying. And they already have a nice mouse model that they're working with. But there wasn't sort of a corresponding in vitro model for a mouse that could be complementary to the amazing capabilities of mouse as an in vivo system for basic biology and disease modeling. So we really wanted to be able to set up, in better directed differentiation systems that allow us to efficiently generate neural cell types from mouse pluripotent stem cells. And that hadn't, for a variety of reasons, that hadn't really been done before. I think, and the big thing was that, when we talk about mouse pluripotent stem cells, most people are referring to, the naive pluripotent state, which is how mouse cells are usually cultured. Whereas with human, of course, it's the prime state. And the difference there is that the prime state, those cells are really ready to differentiate into endoderm, mesoderm, or ectoderm. But the naive state, the cells first have to transit through the formative stage and differentiate into kind of a more of a primed-like state, and then differentiate into defined lineages. So the previous work with mouse directed differentiation mostly started from naive cells and required like an EB or step for the first 2 days to convert those cells into a state where they're amenable to respond to the signaling cues that induce different germ layers. And so, of course, you can also culture mouse cells in the prime state, as Paul Teaser and Ludovic Vallier showed now 15, almost 20 years ago. But that those culture methods hadn't really caught on. And so, people use them from time to time, but mostly people who are studying pluripotency, and they're not used commonly. They were not used commonly as a starting point for directed differentiation, in part because they're difficult to maintain compared to human pluripotent stem cells. And so several labs had shown that if you inhibit Wnt signaling, though, in mouse prime state culture, it actually helps dramatically reduce spontaneous differentiation and make those cultures much more stable. And so we started to play around with those culture conditions a bit, and found basically that we can, if we start a directed differentiation experiment from that mouse prime state, and we modify the differentiation protocols that have been developed for human a bit, we can actually get very efficient differentiation into a variety of lineages, including what we showed initially in a paper in development in 2022, is that we can actually make almost pure definitive endoderm in only 40 hours, starting from mouse prime cells. And so, we had several of our neighbors here at MSK who worked on definitive endoderm, and they were, had mentioned to me, people can't really make definitive endoderm efficiently from mouse pluripotent stem cells. It's not straightforward. And I said, well, maybe we should try this with our prime state cells. And so it turned out it's very efficient. We have this very nice protocol for doing that. And around that same time, Matthias had been working with OTX2 and trying to, he had made some Degron cell lines for OTX2. So you can use a small molecule to degrade OTX2 protein rapidly, which allowed him to be able to start to parse the different roles of OTX2 in early embryonic development. Because of course it's turned on very early during implantation. Then it plays a role in the anterior part of the nervous system. But it's actually also expressed in the definitive endoderm early on, and then slightly later, in the anterior parts of the definitive endoderm. And so it was a very nice synergy, because our definitive endoderm model ended up being a very nice system for his lab to study the role of OTX2 during this transition from prime pluripotency to definitive endoderm, where you do see this strong upregulation of OTX2. And so they're continuing to follow up on that, and they think potentially it plays a role in the anterior posterior patterning of the early definitive endoderm, analogous to its role in the nervous system, but that hadn't really necessarily been appreciated previously. But I think it's a good example of where they didn't necessarily want to have to do human studies to do this, and in vivo it would be very difficult to kind of parse the roles of these transcription factors at these very early developmental stages, where it's difficult to do in vivo studies of the role of a specific gene at E6.75 to E7.25. And so that's where I think these models for mouse can really pair nicely with the ability to do some in vivo work and what's known about mouse developmental biology in vivo. So that's sort of the genesis of that project. And so, it was led by Matthias's lab, but we were able to really provide them with this nice system to be able to study that question.
Yeah, I mean, you talk about the utility of these model systems to dissect the fundamental principles of early neurogenesis and neurodevelopment, and I think there's a utility for differentiation from human pluripotent stem cells or pluripotent stem cells to neuronal cultures or whatever cultures as an appropriate model in some context for studying early development, right? And in my mind, and I've talked about this before on the show as well, I personally think it's the best application for pluripotent stem cell derived systems and organoids in particular, because as we all know, these are inherently immature systems, right? And so perhaps they're best suited to dissecting some of those early developmental principles and mechanisms. So, I mean, would you kind of agree with that in terms of the applications for pluripotent stem cells for modeling early development? Talk a little bit about the utility of PSCs for studying adult versus developmental phenotypes.
Yeah, no, I agree 100%. I think that it actually is relatively difficult to in the mouse embryo between E6.25 and E10. It's extremely dynamic. Most of the lineages are being specified. The embryo is growing dramatically. It's difficult to get embryos out, stage them. It's practically complicated because you also have to sacrifice the mother. And so it's difficult to work with transgenic lines because you're losing the females every time. And so it's non-trivial to do sort of mechanistic and genomic studies of these early developmental fate decisions in vivo in the mouse. And so then I think human pluripotent stem cell models, because they matured more quickly than mouse, I think just because of the intrinsic interest in studying human development, have been used now more and more to study basic questions that actually haven't been that easy to address in the mouse. And so then you get this weird situation where we kind of have done some things in the human and we know about what's going on there. And then we say, oh, it seems a little bit different from what we thought was going on in the mouse, but that's the mouse in vivo. And so then there isn't really like a direct correlate. So it's like having a mouse in vitro model that can be sort of a direct correlate to the human in vitro model, I think really allows you to say more clearly whether something is sort of a human specific biology, or is it, a difference between in vitro and in vivo. And so I think in terms of developmental phenotypes, and basic developmental questions, I think really the stem cell models open up kind of the mechanistic interrogation of these early stages of development, whether it's mouse or human. And so I think, the ability to study mouse early development is sort of an underutilized feature of these stem cell models. And so we've, tried to really highlight how these new stem cell models that we've developed to start from prime pluripotency really allow you to kind of do the same suite of differentiations that are possible in human, do them efficiently and reproducibly, and open up the possibility of having a more to do more mechanistic studies of mouse development in vitro and have this direct correlate to the human pluripotent stem cell models that are available.
Yeah, development is what we talk about on the show, and you know, this is such an exciting time. I say it over and over. Human, as you were just alluding to, is maybe matured a little bit quicker as the interest, but everywhere, And then the basic biology in developmental biology, I would say more than anything, you talk about these embryo models, etc. Fundamental questions are kind of at the fore, I would say. Um, but you're at MSK, Memorial Sloan Kettering Cancer Center, right? Cancer's in the name, and of course, appropriately, this world leader in that realm. Some of the, I would say, maybe the most exciting medical development today, in large part, had its genesis at MSK and Penn, of course. I'm talking about CAR T. Um, but little known fact, MSK is also a powerhouse for developmental biology and stem cells. Like one, I love it because a lot of my collaborators are there. But I wonder, like, is that part of the mission statement? Why do they have such an emphasis in DB there? And a bigger question, if it's who knows, we're just all benefiting from it. If that's the answer, what's it like working there with all these, talk about Lorenz, who's running the center there, Don Way. I mean, there's so many names. Kat, Hadja Tanaka. There's, scores of leaders in the field there. What's it like? Tell me about it.
Yeah, no, it is really an amazing place to work, and I have amazing colleagues in the developmental biology program here, and I think ultimately that derives from the work of our former chair, Catherine Anderson, who of course is an amazing developmental biologist who came to Sloan Kettering in the late '90s from Berkeley, where she had been a pioneer in fly genetics, and unfortunately, so she when I interviewed at Sloan Kettering, she was recruiting me together with Lorenz, but between the time when I interviewed and when I actually came to Sloan Kettering, her health had declined precipitously, and unfortunately, she passed away several years ago now. But she really set up the developmental biology program here and recruited a lot of the current faculty. And so I think the current success of the faculty is really a direct result of her hard work. And so we're all, of course, sad to have lost her. But now, with Lorenz leading the Center for Stem Cell Biology here, he's, the king of 2D differentiation, of course, getting back to the 2D, 3D. One could argue it's 2.5D, but yeah, so having Lorenz's lab here and the Center for Stem Cell Biology that he has set up, we have amazing core facilities with Ting Zhu as well, who's an incredible stem cell core leader. And then, of course, we have this amazing cohort of mouse developmental biologists that often recruited by Catherine Anderson, including Kat Hadjianakis, who's the world expert on early embryonic mouse development, and so it's been tremendously helpful to have those colleagues around as we were trying to set up these new mouse differentiation systems and differentiation models. So yeah, it's really a tremendous place to do developmental biology.
Yeah, absolutely. MSK is not just about the cancer, great DB as well. So thanks so much, Tommy, for catching up with us and taking us giving us a glimpse into your journey from past to present. And of course, amazing work that you did about 15 years ago, and you're continuing to do incredible work at MSK. And before we let you go, we'll ask you a couple of peripheral questions just so our listeners can get to know you a little bit better. So first up, and this is a fun one. If you could answer any single scientific question, regardless of your expertise or chosen field, it doesn't have to be a question in stem cell biology or neurodevelopment. What would that question be?
Yeah, well, I guess I'm tempted to say I'd like to be able to make hematopoietic stem cells from pluripotent stem cells, just shamelessly pander to Daylon's interests. But that's always been an interest of mine. But I'll go with I think it's really amazing how different animals develop. Among mammals, you have mammals of extremely different sizes. So you have whales down to mice and mammals that are even smaller. And I think we really don't have any detailed understanding of how animals' size is controlled. And so it's a developmental biology question, but also the size of organs. Intrinsically, organs know how big they're supposed to be, and that property, I think, how they go about developing and knowing when to stop developing at the right size, I think is always something that's fascinated me about developmental biology. And I think we still don't have a very good understanding of how that works at the molecular level. So I think that's probably what I would say.
Yeah, no, that's definitely a good one. Everything starts as a single cell, one zygote, and eventually that turns into either a Drosophila or a blue whale. So, and the processes that dictate that divergence, I think we can do a better job of figuring that out. That's for sure. And finally, what's the best piece of advice that you've ever been given, professional or not? I like the one that Marius gave you about the Audubon. I do
Yeah.
really like that one, but give us another one.
Yeah. So that was a good one. I guess maybe so that was from my PhD advisor. So maybe I'll say one from my postdoctoral advisor, Michael Greenberg. So he would like to say this is actually originally Arthur Kornberg from Stanford who said this, but don't waste clean thought on dirty enzyme was apparently what Arthur Kornberg always used to say. And so I think that's very good advice, because I think, in science, the reagents that go into your experiments are incredibly important. And I think it's very easy to, start doing things and, starting to make hypotheses based on experiments that weren't, properly controlled, properly set up. And so I think, that was a nice way of distilling that into a short piece of advice where, it really is critical, and especially in the stem cell field, that, the cell lines that you're working with are characterized, the reagents that you're working with are characterized, and that, you're not wasting time, stimulating your cells with something you think is BMP and it's really TGF beta or something like that. So, yeah, I think that, and that's, in the stem cell field, I think there's an increasing appreciation of the importance of all of these underlying factors and how they affect differentiation. And so I think, yeah, I'll go with that one.
Yes, authentication of key resources. Yes, and that, I mean, it takes me back to something you said earlier, apropos to the statement that you made, which I'm really going to take away with me. Was that early experience for you in not? I mean, of course, it's exciting, but the anxiety. I thought you were going to be like, "Oh, we're going to get scooped," but you were just worried that it was an artifact, and I totally get that. But it takes, that's something that I feel like it takes a lot of science maturation to glean. So it's really been a joy talking to you. I really appreciate you giving us that peek into, talk about jumping in the deep end, head first. Not necessarily in your control, but a product of your imagination and sage wisdom in choosing an adventurous and imaginative advisor. Nevertheless, you really have lived a whole science lifetime and continue now to carry on that legacy at MSK, which is pretty strong in its own right. So again, thank you for talking to us about your work and your experience. I think this is a really a great treat for the trainees and the old fogies amongst us listeners.
No, thanks so much. It's great to catch up with you guys.
All right, you guys, that brings us to the end of this episode. Thank you guys so much for joining us. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCellPodcast.com, or by email at info@STEMCellPodcast.com with feedback or to suggest guests. You're going to have a hard time doing better than our guest today, who brought us kind of into the history books, sharing his story about the founding days of direct reprogramming. What a fun chat we had. We got another one for you coming in a couple of weeks. Until then, thank you guys so much for listening.